Welcome back to the deep dive. Today we are tackling what might be the single most complex anatomical restructuring project the human body undertakes.
Building the heart.
Exactly. We're going to trace the cardiovascular system's whole journey from, a simple symmetric tube all the way to that perfectly organized four-chambered fetal pump.
It's an incredible process. I mean, it's just dynamic change where structures migrate, they appear, they disappear. The whole organ has to physically twist itself into the correct orientation.
So our mission for this deep dive is to really distill those slices.
Right. We'll look at the cell populations, that physical twist, the division of the chambers, and the really dramatic flips that happen right at the moment of birth.
And we're going to try and do this in a way that helps you visualize the anatomy without needing to stare at a diagram.
That's the goal.
Excellent. Okay, let's start with the initial blueprint. Before we even have chambers or vessels, where do the building blocks, where do the specialized cells even come from?
So the heart's core structure, it forms from mesenchyme. And this is derived from the midline. Get ready for it.
Yeah.
Splanchnopleuric, coelomic epithelium.
That is a mouthful.
It is. But essentially, this tissue gives rise to the muscle, the myocardium, and the endocardial lining. What's fascinating, though, is that this same initial tissue later gives rise to the epicardium, the coronary arteries, even the connective tissues of the heart.
So that one epithelial sheet is just an incredible multitasker. But the source material, it makes a distinction, right, between where the cells for the primitive heart tube come from versus the cells for the more adult connections.
Exactly. You have two main zones of growth. First is the primary heart-forming field, which you can think of as the cardiac crescent.
Okay.
This is the starting point for the basic heart tube. It forms the bulk of the left ventricle and parts of the atria.
And the second field handles the later, more complicated parts.
Yes, the secondary heart-forming field. This field is located dorsomedial to the primary one, and it's critical. It sequentially adds cells, particularly to the right ventricle and the outflow tract.
Ah, so the parts needed for the pulmonary circulation.
Precisely. So development isn't instantaneous. It's a step-by-step addition of cells from these distinct lineages.
And then there's a third population, one that isn't muscle or lining, but is absolutely critical for the delicate stuff, like the valves and the great vessels.
That would be the head neural crest mesenchyme. These cells migrate specifically to the arterial pole of the developing heart.
And they are essential.
Absolutely essential. They help form the arterial valves and make sure the aortic arteries develop correctly.
Wow.
Without them, the division and the spiraling of the great vessels it doesn't happen correctly. It's why they're implicated in so many severe congenital anomalies.
So we have the building blocks ready. Now for the next impossible task, getting this muscular tube into the right physical position and creating a stable internal scaffold. Phase 1, building the tube in the twist.
Right.
How does the heart even end up in the chest?
Well, a lot of that repositioning is thanks to early embryonic head folding.
Head folding.
Yeah. Initially, the heart tissue is actually cranial. It's above the developing foregut. As the embryo folds, this position completely reverses. The heart tube gets pulled down, ventral to the foregut.
And that creates the space for it.
It creates the future pericardial cavity.
Okay, so once the tube is formed and it's in position, I understand there's a kind of temporary support structure holding it in place.
There is. It's called the dorsal mesocardium. The easiest way to think of it is like the heart's temporary mesentery. It attaches the back of the heart tube to the dorsal pericardial wall.
But it doesn't last long.
No, it breaks down pretty quickly.
Yeah.
And that empty space it leaves behind becomes the transverse sinus of the pericardium, which is a key landmark in the adult heart.
Okay, here's where it gets really interesting structurally. The creation of internal scaffolding using what was, historically called cardiac jelly.
Ah, yes, the cardiac extracellular matrix. It's central to this next step. What happens is a pretty profound change in cell identity. Signals like BMP2 and TGFF coming from the surrounding heart muscle, the myocytes. They induce a subset of the endocardial cells, so the lining of the tube, to undergo epithelial to mesenchymal transition, EMT.
Hang on. So the cells that line the inside of the tube, which are fixed endothelium, they transform into mobile proliferating mesenchyme.
Precisely. It is incredibly unique. This is mesenchyme derived from endothelial lineage, not the typical mesoderm. These mobile cells then invade that matrix, that cardiac jelly, and they form the endocardial cushions.
And these cushions are vital.
Oh, absolutely. They act as temporary valves to regulate flow in that simple tube, and later they provide the bulk of the tissue needed to complete septation across all 4 chambers. They are both the scaffolding and the material for the doors.
So we have a straight tube, it's supported internally, and now it's ready for its most crucial movement,
The loop.
the twist of fate.
Describe that physical maneuver for us, the one that sets up the whole left-right alignment. The heart tube starts as an inverted Y. The 2 legs at the bottom are the venous inflow, the future atria, and the single stem at the top is the arterial outflow.
Okay.
This whole structure lengthens, and then it begins a rightward and ventral looping process. Imagine the tube literally bending on itself, kind of like not being tied.
And that movement is what places the ventricles correctly.
It's everything. It ensures the developing left ventricle is positioned on the anatomical left, and the right ventricle moves to the right. If this loop is misaligned, well, the subsequent septation will fail.
That structural choreography sets up phase 2, compartmentalization. Let's start at the back end, tracing the venous inflow tract.
Okay, so initial venous return is symmetrical. It's flowing into these bilateral structures called the sinus venosus from the umbilical, vitelline, and cardinal veins.
But it becomes asymmetric very quickly.
Dramatically asymmetric. The right sinus horn enlarges and it gets absorbed into the right atrium, forming that smooth-walled posterior section, the sinus venerum.
And the left side becomes almost vestigial, right?
It shrinks down, significantly. Yeah. The left sinus horn pretty much just becomes the coronary sinus, which drains the heart muscle itself. And uniquely, it retains its own myocardial wall.
And the original valve leaflets.
They regress, but they leave remnants we can see in the adult heart. The crista terminalis up top, and the valves protecting the IVC and coronary sinus, which are the Eustachian and Thebesian valves.
Okay, let's move to atrial septation. This is that sophisticated two-septum, two-foramen system. It's this beautifully engineered design for the fetal shunt.
It really is. It starts with the septum primum. This is a thin crescentic membrane that grows down from the atrial roof toward the AV cushions.
It leaves a gap.
It leaves a critical gap at the bottom, the foramen primum, for shunting. But here's the clever part. Before it even fully fuses, the superior part of that septum undergoes apoptosis programmed cell death.
So it punches a hole in itself.
It punches a new hole in itself, the foramen secundum, to maintain this shunt.
And then the second septum comes in to create the actual functional door.
Exactly, the septum secundum. It's much thicker, more muscular, and it develops to the right of the primum. But crucially, it never completely divides the atria. It just acts as an overlapping shield.
And the gap between them.
The gap between the bottom edge of the secundum and the top edge of the primum establishes this oblique one-way pathway. That is the foramen oval. It ensures the septum primum acts like a flap valve, directing oxygenated blood from the high-pressure right atrium straight across to the left.
That flap valve mechanism is truly elegant. But if that choreography is mistimed, that's where we get atrial septal defects.
Right. The most common type is in the foramen secundum region. Either the septum secundum shield is too short, or the hole in the primum is just too large.
I see.
We also see sinus venosus defects. They're high up near the entrance of the venae cavae, and they can be clinically significant because they're often associated with anomalous pulmonary vein drainage.
Okay, now for the big one. Separating the ventricles and the great outflow arteries. How does that single tube become 2 distinct pumps?
Ventricular separation starts with the muscular ventricular septum. Its crest is actually the oldest primary myocardium. The ventricles then kind of expand or balloon out around this fixed crest.
But that doesn't close the hole completely.
No, to close the final communication, the interventricular foramen, you need a fusion of 3 structures. That muscular septum crest, an extension from the dorsal AV cushion, and most importantly, the outflow cushions.
And the division of the single outflow trunk, the truncus arteriosus, into the aorta and pulmonary trunk.
This relies entirely on the formation and fusion of the spiraling outflow cushions, along with that input from the neural crest mesenchyme we mentioned earlier.
And the spiral is key.
The spiral is everything. That's how the aorta and pulmonary trunk end up wrapping around each other and connecting to the correct ventricles. This whole complex forms the aortopulmonary septum.
And that spiral division is where so many severe defects can originate?
Precisely. If those cushions fail to fuse completely, you get persistent truncus arteriosus, where the outflow is just a single vessel. Or, if they fuse without the proper spiral, you get transposition of the great arteries, where the aorta connects to the right ventricle and the pulmonary trunk to the left.
And of course, VSDs.
Ventricular septal defects, yes. very common. They can be near the central cushions, those are perimembranous VSDs, or you can have multiple small muscular defects, sometimes called a Swiss cheese septum.
That brings us to phase 3, the supporting cast, how the heart gains its own power system and blood supply, and how the whole systemic circulation gets reorganized.
Right, the infrastructure.
Let's start with the muscle itself. How does it differentiate to create the electrical system?
The developing myocardium differentiates into two main types. First, you have the primary myocardium. This tissue retains its intrinsic rhythmicity, its automaticity, but it has slow conduction speed.
So this is the pacemaker tissue.
It is. It differentiates entirely into the specialized electrical system. The SA node, the AV node, and the bundle of His and Purkinje fibers. The entire electrical wiring is muscle-based.
And the second type does the actual heavy lifting.
That's the working myocardium. This tissue is all about high contractility and fast conduction. It makes up the bulk of the atrial and ventricular walls. It's what you need for that rapid, forceful, synchronized contraction.
How does the heart get its external layering and, crucially, its own blood supply?
The heart surface gets covered by the epicardium, which actually develops from a little structure called the proepicardial organ near the sinus venosus. This tissue migrates and spreads across the heart.
And the coronaries?
The coronary arteries are really unique. They develop by vasculogenesis, meaning they form in situ, right there,
Right.
as a plexus of new vessels within the subepicardial mesenchyme.
So they don't branch off the aorta initially.
No, that plexus only establishes a connection to the aortic sinuses later on during that crucial phase of outflow tract septation.
Now we turn outward to the systemic vessels. Arterial remodeling seems, well, relatively straightforward compared to the veins.
It is simpler, certainly. The bilateral dorsal aortae fuse early, forming the definitive descending aorta. A key point here, though, is the ventral splanchnic arteries. They start as paired vitelline vessels, but they consolidate to become the unpaired gut trunks we know.
The celiac, SMA, and IMA.
Exactly. The celiac trunk, superior mesenteric, and inferior mesenteric arteries.
Okay, now for the grand challenge, the venous system. It's an asymmetric labyrinth of change, defined by a huge reorganization and a shift to right-sided dominance.
It's truly a complex reshuffling. The early symmetric cardinal system, it just has to undergo a massive change. If you try to visualize the inferior vena cava, you have to understand it's not built whole. It's a composite vessel, a biological jigsaw puzzle.
A puzzle made of what pieces?
Segments of the right subcardinal vein, the right supracardinal vein, and the right hepatocardiac channel. These different shifting vessels fuse sequentially to form that final right-sided main return channel.
Which explains why anomalies there are often related to missing or duplicated segments.
It does.
And the portal vein system, that's intricately tied to the liver and the gut.
That's right. The portal vein develops from a complex series of cross-connections and anastomoses among the vitelline veins that surround the developing duodenum. Because the duodenum moves and elongates, these connections form a pathway often described as a figure of 8.
And that's what integrates drainage from the spleen and the gut.
Right, from the superior mesenteric and splenic veins into the liver's circulation.
We've finished building the pump and the pipes. Now for phase 4, fetal circulation and the postnatal switch. Let's ask you, the listener, to visualize the single source of oxygen. The placenta. High oxygen blood returns via the single left umbilical vein.
And that blood is immediately shunted to prioritize the most vital organs. It reaches the liver, but mostly bypasses it via the ductus venosus.
A shortcut.
A direct shortcut into the inferior vena cava. This high oxygen stream then flows into the right atrium.
And this is where that elegant atrial septation comes into play, ensuring that precious oxygen goes where it's needed most.
Exactly. The pressure and the way the blood streams guide it right across the right atrium, through the flap valve of the foramen ovale, straight into the left atrium and left ventricle.
So it's preferentially delivered to the heart and the brain.
The heart muscle and the aortic arch for the fetal brain, yes.
The rest of the systemic return comes from the head and upper body via the SVC. That's less oxygenated. Where does that go?
That blood streams into the right atrium, down into the right ventricle, and out the pulmonary trunk. But since the lungs are collapsed and have extremely high vascular resistance, most of it, up to 2/3 of the cardiac output, bypasses the lungs entirely.
Through the ductus arteriosus.
Through the ductus arteriosus.
Yeah.
This shunt, a remnant of the left 6th pharyngeal arch artery, connects the pulmonary trunk directly to the aorta. It's a beautifully balanced, high-pressure system for life in utero.
But everything changes at the first breath.
The transition is dramatic, almost instantaneous. The lungs expand, and pulmonary vascular resistance just plummets.
So blood rushes to the lungs.
It does. And this immediately causes the pressure in the left atrium to soar, surpassing the pressure in the right atrium. That pressure difference slams the septum primum shut against the septum secundum.
A functional closure of the foramen oval.
A rapid functional closure.
And the arterial shunts, the ductus arteriosus, they close because of a biochemical shift.
Two major chemical changes. First, increased oxygen tension in the blood acts directly on the muscle. Second, a sudden drop in circulating prostaglandins PGE2, the molecules that kept the ductus open, causes the smooth muscle in the walls of the ductus arteriosus and the umbilical arteries just contract rapidly.
And functional closure happens within hours.
Usually, yes.
These temporary fetal structures then undergo fibrosis and turn into the ligaments we recognize in the adult body.
A final list of fates for you. The ductus arteriosus becomes the ligamentum arteriosum. The umbilical arteries become the medial umbilical ligaments. The ductus venosus becomes the ligamentum venosum.
And the umbilical vein.
And the single umbilical vein becomes the ligamentum teres hepatis, the round ligament of the liver.
The failure of these closures is what constitutes the majority of congenital heart disease, which occurs in about 8 out of every 1,000 births.
That's why that postnatal switch is so critical. A patent ductus arteriosus failure to close within 72 hours is especially common in preterm neonates, often because there isn't enough oxygen tension to trigger that muscle contraction, which leads to catastrophic shunting from the aorta back into the pulmonary circuit.
So if we synthesize this entire journey, from a simple tube to a critical rightward loop, meticulous septation guided by cushions, and finally the complete remodeling of the circulation, it really speaks volumes about the necessity of precise timing in embryology.
Absolutely. The progression is defined not just by what is being built, but by what is simultaneously being dismantled, absorbed, or transformed. It's a dynamic, ever-changing spatial relationship.
And here's a final provocative thought for you to mull over. What's so fascinating here is that heart development isn't just a purely genetic cascade, it's profoundly mechanical. The source material suggests that hemodynamic forces, the actual pressure and shear stress of the blood flow itself, are critical for vessel wall thickening, for regulating caliber, and even for guiding the morphogenetic movements of the heart.
Right.
So consider this. How much of the final adult heart anatomy is predetermined by genes, and how much is physically sculpted by the blood that flows through it? That's a deep dive for another day.
An excellent point. It really underscores the dynamic nature of embryology and physiology working in tandem. Thank you for joining us as we navigated the development of the heart and circulation.
Until next time, stay curious.